Hello, students. Welcome to the class of pyrimidine synthesis and catabolism. We all know that purine and pyrimidine are a part of nucleic acids and they form the DNA and our RNA. Number of amino acids are synthesized into purine and pyrimidine nucleotides. They are important as energy currency of cells, that is the ATP, and in some cases it might be even GTP. UTP, which is the uridine triphosphate, is the source of energy for activation of glucose and galactose. CTP, that is your cytosine triphosphate, is the source of lipid metabolism. Apart from this, we have our AMP, which is part of a coenzymes like NAD and coenzyme A. And nucleotides are part and parcel of our nucleic acids, which forms our DNA and RNA molecule. Actually, we don't require these in diet as we produce them in de novo. So, we shall today look into the synthesis of pyrimidine and catabolism. So, these are the pyrimidine structures what you are observing on your screen. That is the cytosine, uracil, and thymine. So, uracil is the analog of thymine in RNA. The raw materials which we require in the synthesis of pyrimidine are glutamine, aspartate, tetrahydrofolate, folic acid, or vitamin B9. And in this slide, you can see how the synthesis of a pyrimidine occurs. So the first step, glutamine, two molecules of ATP and carbon dioxide are utilized by our cells and converted into carbamyl phosphate by the enzyme carbamyl phosphate synthase 2. You know that you have already heard the name of this enzyme, carbamyl phosphate synthase 1, which was present in the conversion of ammonia into urea in the urea cycle. It was present in the liver mitochondria, whereas this is CPS2, which is present in the cytosol and it converts the glutamine, ATP, and carbon dioxide to carbamyl phosphate, wherein the glutamine is converted to glutamate. And here you can see that some of the arrow marks throughout the slides you can see they are red and some are green. Please remember the red are the inhibitors of the enzyme and the green are the promoters or activators of the enzymes. I shall not be dealing with the regulation of the enzyme herein. The carbamyl phosphate is then converted into carbamyl aspartate in the presence of aspartate transcarbamylase in the presence of aspartate enzyme. That is, the aspartate molecule is being added into carbamyl phosphate, which then converts into carbamyl aspartate, which in turn gets converted into orotic acid in the presence of enzyme dihydroorate dehydrogenase. The orotic acid is the first pyrimidine nitrogen base to be formed. That is, it is the nitrogen group to be formed, not the complete base. Simultaneously, you can see that pentose phosphate pathway, which occurs in your body, produces ribose 5-phosphate, which is converted into 5-phosphoribosal 1-pyrophosphate. In short, it is termed as PRPP in the presence of the enzyme PRPP synthase. The PRPP then combines with orotic acid to form orotidine 5' monophosphate, which is known as OMP. The OMP is then converted to uridine monophosphate in the presence of OMP decarboxylase with the removal of carboxylase group, or sorry, the carbon dioxide. The UMP is then converted to uridine diphosphate and deoxyuridine diphosphate in the presence of the enzyme ribonucleotide reductase, and then to deoxyuridine monophosphate, which in turn gets converted to deoxythymidine monophosphate in the presence of the enzyme thymidylate synthase, which is a very important enzyme which requires the cofactor N6N10 methylene tetrahydrofolate, which gets converted into tetrahydrate folate. This N6N10 methylene THF in turn is regulated by dihydrofolate reductase, which has not been mentioned in the slide. But do remember, most of your chemotherapeutic drugs are targeted against this dihydrofolate reductase. So it acts on the folic acid, that is your thymidylate synthase is being inhibited. So the DTMP is then converted to DTDP and DTTP, that is deoxy tiny triphosphate, which then proceeds into the DNA formation in the presence of the enzyme or DNA synthesis. And the enzyme is known as DNA synthase. So, this is the main step in the DNA synthesis. We also have the side reaction, as you can see here. UDP is converted to UTP and then into CTP, wherein the UTP gets converted to CTP in the presence of the enzyme CTP synthase in the presence of ATP and glutamine. The UTP and CTP then in the presence of the enzyme RNA synthase can get converted into RNA. The CTP form is into CDP and deoxy CDP and deoxy CTP, which again can produce the DNA along with DTP. So this is about your pyrimidine synthesis. Looking into your pyrimidine catabolism, we know that catabolism is breaking down of your components into simpler forms. So this is your DNA. And what are the pyrimidines? You know it is the cytosine and thymine because we are speaking about DNA. The cytosine can get deaminated to uracil, that is by the removal of ammonia group. Both the thymine and uracil can enter into the pyrimidine salvage pathway. The salvage pathway means the salvage pathway in which the biological product is produced from the intermediates in degradative pathway of its own substances are known as salvage pathway. So the uracil is converted to dihydrouracil thymine to dihydrothymine by the rate limiting step, that is an enzyme dihydropyrimidine dehydrogenase, which actually controls the entire pyrimidine catabolism. The dihydrouracil or dihydrothymine is produced by the removal of the hydrogen group from the NADPH. Dihydrouracil in turn is converted to N-carbonyl beta-alanine and dihydrothymine is converted to N-carbonyl amino isobutyrate. This in turn are converted, that is your N-carbonyl beta-alanine is converted to beta-alanine with the addition of water in the presence of the enzyme beta-uridopropionase. and your dihydrothymine is converted to N-carbamyl amino isobutyrate. So both these reactions are catalyzed by beta-uridopropionase, your beta-alanine and beta-amino isobutyrate forms a common byproduct, ammonia and carbon dioxide. Ammonia is, as you all know, is toxic to humans or any other animals, and it can be converted into less toxic urea by the urea cycle, and it can be excreted in the kidney. The beta alanine is converted to acetyl CoA, and beta amino isobutyrate is converted to succinyl CoA. Again, as you all know, acetyl CoA and succinyl CoA are part and parcel of your TCA or Krebs cycle. So they form the intermediates of your TCA or your Krebs cycle, or your even which is called as a citric acid cycle, wherein the main aim of your reaction is to produce once again the ATP and the required metabolites. So this we conclude a class on the pyrimidine catabolism and pyrimidine synthesis. Thank you.